Hello, everybody. Welcome back to Creation Myths. Today, we are doing a literature review of an actual scientific paper published in an actual real journal by Creationists. That's something that doesn't happen very often. So we're going to check out this paper. It was published back in the fall of 2021, so it's pretty recent. So we're going to look at this paper and we're going to see if it stands up to scrutiny. Before we go any further, if just in the chat for the people watching and listening, if the sound is okay, just let me know. If you can't hear me, let me know. Just let me know how everything is before I go any further. And if everything looks good, then we can get right into it. So I'm going to start with a discussion, and if I see anybody say anything in the chat that there's a problem, then I will try to address it. But let's get right into it. So I'm going to show you the paper. And then I want to talk a little bit about kind of the whole thing of creationists publishing real science papers. So let me pull up the paper, but then we'll talk a little bit about this kind of genre of publication before we get into the actual kind of nuts and bolts of what's going on in this exact paper. So here's the paper we're going to be talking about. This is on the waiting time until coordinated mutations get fixed in regulatory sequences. This is by Ola Hössjer, Günter Bechly, and Anne Gager. I think that's pronounced Gager. That's how I've heard it before. I apologize to Dr. Gager if that's incorrect. So this was published in the Journal of Theoretical Biology. I'll talk about that a bit later on, but that's a journal where you get some kind of mess stuff. And oftentimes, this is not the first time creationists have been published in this journal. And it's often a way where you can get kind of something published without it being reviewed by the specialists in that particular field. So this is not the first time that this journal has hosted a creationist set of authors essentially making a stealthy creationist argument. So I'm going to take this down because we're going to talk about this in quite a bit of detail. We'll get there in just a few minutes. But before we do that, I want to talk kind of more broadly about this. So this, as far as I know, is the only real peer-reviewed paper that creationists put out in 2021. It's possible I missed a few, but mostly they don't even try to publish in the actual peer-reviewed literature. You do see a few examples of this, and the examples that you see tend to be pretty well known because creationists keep citing the same 3 or 4 papers over and over and over again because those are the only ones they can go to. So you have the Douglas Axe paper from 2004 on the frequency of functional protein sequences. You have Bechly and Snoke 2004 on the evolution of an irreducibly complex protein interaction. You've got, of course, Carter and Sanford 2012, my favorite terrible paper of all time on the H1N1, the 1918 H1N1 pandemic and its evolutionary trajectory. I did a video on that a while back, a couple years ago now at this point. So you can check that out if you want. You also see a lot of creationists publishing in kind of the in-house journals. I'm going to put that in quotes, the in-house journals. Those don't count. Those are not real journals. So that's like Answers Research Journal. That's from Answers in Genesis, of course. You've got Biocomplexity from Discovery Institute. These are propaganda outlets that are masquerading as peer-reviewed publications. They're not actually peer-reviewed publications, right? These are not real journals. They're not sending them to specialists in the field to review. They are kind of closed circle type things, right? So within Answers Research Journal, it's all the AIG people, reviewing each other. With Biocomplexity, if you look at the lists of reviewers and stuff and the editors, it's all just Discovery Institute people. You look at the authors, it's Discovery Institute people. It's just a closed circle within their organizations. I mean, there's no actual peer review going on in these publications. They're essentially glorified blogs, is what they are. So this paper from 2021, this waiting time problem paper, I'll put that in quotes, this waiting time problem paper, because the waiting time problem's not a real thing. This paper got a lot of publicity. So you saw it, for example, on the Evolution News blog from Discovery Institute. You saw it in a couple other creationist outlets saying, "Hey, look, this is a this is a real thing." In fact, just a couple of weeks ago, Discovery Institute kind of put out a thing of the 3 biggest developments in intelligent design in 2021. And this paper, the publication of this paper, was one of them. So that's kind of where this paper fits, kind of in the broader context of creationists trying to promote their ideas. It's this kind of thing where they're putting it out into the actual peer-reviewed literature, sometimes in journals that are not, optimal for the thing that they're talking about. Another example of that is the 2012 H1N paper, which was not reviewed by virologists. It was not reviewed by evolutionary biologists. Like, it wasn't in a journal on evolution. It wasn't in a journal on virology. It was, I think that was also Journal of Theoretical Biology, actually. So you see that kind of thing sometimes. So let me bring this paper back up, and we can talk specifically about what we're doing in this paper. So the central claim in this paper is that there is this thing called the waiting time problem. And this is the idea that it takes too long to get a specific set of mutations or alleles together in a genome. So if you need a certain set of mutations in order to acquire some trait, well, it takes so long to get all those things together that it's functionally impossible for evolutionary processes. This argument is basically one answer that creationists have put forward to the what's the limit question that people ask, because you see creationists will accept, everyone accepts microevolution, everyone accepts adaptation, right? Everyone accepts that allele frequencies change within populations. Well, if you can look at mutations and you can see that it's just mutations that are responsible for the differences between, maybe it's dogs and wolves, maybe it's dogs and coyotes or foxes, or maybe it's dogs and other members of carnivora, right? Well, if it's just changes to the genome and regulatory changes and whatever, right? If we can document the types of changes that separate different organisms from each other, the question that, people who accept evolution, mainstream scientists will ask of creationists is, okay, well, if you can accept that a common ancestor can get you A and B, or A, B, and C, why can't a common ancestor in the more distant past gets you A and Z, right? Why can't you get from A to W? And this waiting time problem is one attempt to answer that question. Some creationists will say that there are specific types of traits that cannot evolve, right? So you got like irreducible complexity from Michael Bechly, right? There are specific traits that cannot evolve if they meet certain criteria, right? Now, the other approach to that, and that approach has failed. Like, that doesn't, like, irreducible complexity doesn't work. You can't just draw a line and say, like, beyond this point, it's too complex, evolutionary processes can't do it, because whatever standard creationists have come up with has actually been, like, experimentally shown to be not valid, right? We've shown experimentally that you can evolve irreducibly complex traits very quickly, right? That's been done. So another answer to this question is, well, it's not about the specific characteristics of the trait, it's about the number of mutations, right? Or the number of differences between, your common ancestor and your descendant, between the ancestral trait and the derived trait. It's the number of genetic differences that matter. And beyond a certain number, it just takes too long via evolutionary processes to acquire all of those differences, right? We just don't have the time in a large enough population, and the genome's too big, right? We just have too much sequence space to sample to achieve these specific genomic characteristics that we need. So the answer to the question of what's the barrier to evolutionary processes, according to the waiting time problem, it's not a specific trait or a specific type of trait. The barrier is time and the number of changes that are required within a lineage, right? So what these authors are doing is they're constructing a population model where they're aiming to show that this waiting time problem is an actual problem for evolution, right? That's what they're doing. Now, they don't explicitly say this. In the greatest traditions of creationists publishing their stuff in real peer-reviewed journals, they're not actually making the argument that they want to make, right? So what creationists do with this kind of thing is they publish the paper, then they turn around and go, see, aha, this is evidence for intelligent design. Or Carter and Sanford with the H1N1 paper, see, aha, this is evidence for genetic entropy. But they're not willing to make that argument in the peer-reviewed literature. They're willing to futz around the edges, right? And like come up with a model that maybe shows this, or finagle the influenza mutation data to, maybe it shows that, right? but they're not actually willing, apparently, to make the central argument that they want to make. Right. So with that out of the way, let's talk about this actual paper. Let's get into this actual paper now. So what these authors did is they constructed a model, and they said, okay, given a set of characteristics, a set of parameters for our model, and a set of characteristics for this experimental population in the model, how long would it take to generate a specific regulatory sequence, right, in this hypothetical genome? Now, they set a series of parameters for the population in their model, and you're going to see as we go through them, many of you are going to hear these parameters and think, that doesn't sound quite right to me, and you are going to be correct when you think that. So the populations that they describe are haploid and asexual, right? So non-recombining, they have one copy of the sequence in question per individual. They have in their model a specific target sequence, right? So they have, okay, so we have a starting point and we have a target end point, right? And the question is, how long does it take to get from point A to point B, right? There's a specific target sequence. And the mutations that you would use to get from point A to point B must occur sequentially. So basically the way they describe it is mutation number 1 occurs, fixes, number 2 occurs, fixes, right, in sequence. And they also account for back mutations. So it'd be like number 1 occurs and then goes away. Then number 2 can occur, but then you're missing number 1. You got to get number 1 back, right? So it's forward and back mutations in sequence within this haploid non-recombining population as you move from an ancestral sequence to a target functional sequence. So having very briefly described that model, I'm sure many of you listening can identify some of the errors there. I hope many of you can identify some of the errors in there. So let's go through them. This is just the abstract. As I was going through this, I just highlighted for myself some of the relevant stuff. So I'm going to go all the way down to the discussion here. So we'll go all the way down to page 19. Here we are. So the first thing I want to point out is this paragraph right here. What the authors say is that it is reasonable to believe that recombination between 2 parental regulatory sequences can be ignored, since both of them are well approximated by the corresponding consensus regulatory sequence. In other words, within the population, there's maybe some variation in the sequence that you're evolving, but everyone's close enough to a consensus sequence, kind of an average of the population, that we're going to discount recombination. Now, you can't just ignore recombination, right? The calculation they're trying to do is how long does it take to get mutation A, B, C, D, however many mutations, right? They talk about how in here, how you can, change the number of mutations that are required. However many number of mutations it takes, they're talking about how long does it take to get all of them together. Well, one way you combine mutations within a population is through recombination. So you can't discount that as a factor when you are considering the amount of time it would take. This is true even for their haploid asexual population. Things that are haploid and asexual still do recombination. Okay, so think about a bacterial population. Think about prokaryotes, right? They're haploid, they're asexual, they just binary fission, they divide really fast. Well, they're still recombining with each other. They're not doing sexual reproduction, right? They're not recombining as part of their reproductive process. They're not finding a mate and swapping and doing crossing over, but there's still horizontal gene transfer going on between the individuals in that population. So even according to the parameters of their model, even if you maintain haploid and asexual, you still can't discount recombination as a factor that is going to speed up the rate at which you're going to arrive at your target sequence. That's problem number 1. And honestly, we could stop there. That right away is enough to invalidate whatever claims they're going to make based on this model, right? Stop right there. We could be done. We're not, because there are more problems, but we could stop there if we wanted to. So now let's hop back up a little bit to page 17, because the second problem here is right in this line. They say, assuming that the population is small enough to warrant a fixed state approximation whereby all individuals have approximately the same regulatory sequences. What they're talking about here is they're saying at any given time, there is little, if any, variation within this population. And basically, you have to do mutation number 1 And then if it fixes, then you can have mutation number 2. And then if that one fixes, now you have the 1 and 2. Now you can have mutation number 3 occur in a sequential fashion. That's not how evolution works. Evolution is a massively parallel process. You have a population of individuals. All of those individuals are, if you can think about it this way, they're trying out all different mutations all at the same time, right? So this goes hand in hand with the recombination. You don't have just one sequence within the population at any given time. You have lots of sequences in that population at any given time. So they're not just like, okay, is this the mutation we need? No. Okay, try the next one. Is that the one we need? No. Okay, try the next one. Is that the one we need? No. It's dozens, hundreds, thousands, billions, however many individuals are in that population. They're all mutating on their own lineages and their own trajectories. And the authors are just discounting that. Basically, they're flattening all that variation that's occurring to a single consensus sequence at any given time in that population. You can't do that. That's not how evolution works. That's not a valid approach. There's no populations that actually work like that in the real world. So if you're trying to describe how this so-called waiting time problem is a real problem in the real world, a population like this is not appropriate for that. That's not how you're going to do it. So then the third problem, and this is the big problem. Like, either of the two things I've already described invalidate the central claims here, but this one, this is huge. This is like big, giant red flag. There's no way this should have gotten through peer review if any actual biochemists or evolutionary biologists or anybody like actually reviewed this paper, because this third error is the big one, and it's completely inescapable and invalidates everything about this paper. And the third problem, and we're going to go back down to page 19, but you can see it throughout here, is that they have analyzed the waiting time for a fixed and pre-specified target sequence. Fixed and pre-specified. Evolution does not have a fixed and pre-specified target sequence. It's not how evolution works. Basically, what the authors do here is they start with a functional sequence, right? Say, this is the thing we want to get. We know that this sequence is functional. And then they work backwards from there, saying, okay, how do we get to that functional sequence from some hypothetical ancestral sequence? Okay? This is an extremely common error when creationists talk about the waiting time problem. This is the exact same error that Douglas Axe made in his 2004 paper when he was looking at the frequency of functional whatever protein sequences. He does the same thing. He has a pre-specified target. Michael Behe has committed the same error, where he's dealing not with the whole universe of functional sequences, but with specific target sequences, saying how long will it take to get this thing? Because this waiting time problem is related to Michael Behe's edge of evolution kind of hypothesis, right? It's the same basic argument of it just takes too long to get changes of a certain degree, right? The math that when creation scientists do this, the math that they do involves a fixed and pre-specified target. Evolution does not have targets, right? We're not trying to find the specific enzyme that exists today, right? There's variation, and like the enzyme that does the thing approximately, whatever variant of it appears, and then it's optimized through natural selection over time. Now, so the relevant question that creationists need to be dealing with is not how do we get this one sequence? They're ignoring that there are lots of sequences that will do the same thing approximately equally well. The question they should be asking, and the question that is not asked here, is how long does it take to get any functional sequence of a particular flavor, right? How long does it take to get a polymerase? How long does it take to get a disulfide bond? Whatever it is, it's not this specific one. It's how long does it take to get anything that will work for that particular thing. And the interesting thing is that we have data on this question. This is not some unknowable thing. People have done work on this. So I'm gonna put down the one paper here, and I'm going to pull up a different one because I'm not the first person, as you can imagine, to address this this waiting time problem paper, it was addressed. Just one second. Let me just pull up a different paper here. So I'm just going to share this other one. I accidentally closed it. So here we go. So this is a different publication. This is on page 6 here. This is Yona et al. 2018. Random sequences rapidly evolve into de novo promoters. That's an interesting title, and it seems relevant to what we were just talking about. So this is actually from a piece on the panda's thumb. I forget the name of the author on the panda's thumb piece, but I have it linked down below in the description, so check that out. The author goes into some of the stuff I talked about here. Also talks about other aspects of the waiting time paper that are a problem. But I want to just finish today by talking about this paper specifically because it does actually address this problem. What the authors did in this paper is super cool. So they took some E. coli and they knocked out the lac promoter. So anyone who's had like first year genetics, you probably have heard of the lac operon. Well, lac operon has a promoter. It's about 100 base pairs long. And what they did is they knocked it out. And we can just scroll down here and show you the experimental procedure. What they did is they knocked it out, right, and replaced it with random sequences of the same length. So just random sequences of 103 bases. And then let's just see. And then we'll grow those bacteria just on lactose. It's called a minimal medium, right? Because it's just one nutrient, lactose. And let's see if there's anybody that can survive in that lactose-only environment with just the random sequences for the promoter rather than the correct promoter. Well, it turns out, yeah, a lot of things can survive. A lot of things do just fine with those random sequences. So what they found, we'll go down here to look this figure right here. We'll go down here. What they found was some random sequences are actually able to directly function as the lac promoter, right? Some of those random sequences that they did could directly function as the promoter. And a whole lot of them, if you look at this second part of the figure down here in blue, this is the distribution after one mutation. So this is one mutation different from the random lac operon sequences. Sorry, everybody, one second. So what we have here, this line represents the wild type level of expression. This is 0. So you can see not just did one mutation recover some promoter functionality in the majority of these sequences, for some of them, it met the wild type and it exceeded it by 50%, 100%, 150%. It was actually a higher activity promoter with a random sequence and a single mutation. And the last figure I'll show you is this one, just looking at the distribution here. So this is the number of mutations needed to become a functional promoter. Some of them functioned out of the gate. Some of them functioned with 1. Most of them functioned with 1. Some required 2 or more. So the central premise of the Creationist paper here is wrong. That, it takes too long to get a functional sequence. The reason they're wrong, the central reason they're wrong, is they are aiming, if you were doing it for the lac promoter, they're aiming for the existing lac promoter. But it turns out a lot of sequences will work equally well as a lac promoter. Just randomly generated sequences followed by mutations. Turns out those work really well as a lac promoter. And by limiting their analysis to a single target sequence, the authors completely ignore that. And that is the central reason why the waiting time paper by Ola Hössjer, Günter Bechly, and Ann Gauger is wrong. The central thing that they're trying to show, that the waiting time problem is actually a problem for evolution, is wrong because the version of evolution that their model utilizes bears no resemblance to evolution in the real world. So to briefly summarize, they exclude recombination entirely. They treat it as a sequential rather than parallel process. And most importantly, they have a pre-specified target sequence rather than recognizing that many sequences will do a job approximately equally well. And once you arrive at any one of those sequences, natural selection and mutation can get you to any of the rest of them because they're just sequence-based. They're connected via mutations. So that's my brief look at let me get the title exactly right for everybody. That's my brief look at On the Waiting Time Until Coordinated Mutations Get Fixed in Regulatory Sequences by Hössjer, Bechly, and Gauger. This is from the Journal of Theoretical Biology from I think it was from the fall of 2021. And the idea that this is a pro-creationist paper or a pro-ID paper that somehow bolsters the case for the waiting time problem is just ludicrous. The model that they use is completely unrealistic and useless in terms of actually learning about real evolutionary processes. So that is what I have to say about that. I hope everyone enjoyed that, and I hope now everyone here knows when this paper comes up, because creationists love to promote this paper. When it comes up, you can explain exactly why it is wrong. Thank you for watching, everybody. Have a great night. If you haven't already hit like, just do that for me before you go. Or hit dislike. That's fine, too. I don't care. The algorithm treats it the same. That's fine. And if you haven't subscribed, please do subscribe, because this is the kind of stuff we do here. We look at creation science, and we take it seriously and see that it's wrong all the time. That's what we do here on Creation Myths. So thank you, everybody. Have a great night. I hope I'll be back soon with some other fun stuff.